Welcome back to the Science Hub SKB. Working like a Scientist, Part 6. The objectives of this part of the course are: what is a variable? What is an independent variable? What is a dependent variable? And what is a controlled variable? What are variables? In science, a variable is anything that can change or be changed in an experiment. There are three main types. A, the independent variable. This is the one you change on purpose. B, the dependent variable. This is the one you measure or observe. And C, the controlled variables. These are the ones you keep the same so the test is fair. Types of variables. Independent variable. This variable is also called the manipulated variable. This is the factor that is intentionally changed or controlled by the experimenter to observe its effects. Dependent variable. This variable is also known as the responding or measured variable, which is the factor being observed and measured. It changes in response to the independent variable. And the control variable. These are factors that are kept constant throughout the experiment to ensure that any observed changes in the dependent variable are due to the manipulation of the independent variable alone. Okay, so let's try some exercises now. You are given 10 situations. Determine for each situation what the independent, dependent, and control variables are. The first one is done for you. Our first investigation states, a scientist tests how different amounts of sunlight affect plant growth. So what we're manipulating here would be the amount of sunlight. The independent variable is the one that you purposely change. Now, as we move on to the dependent is the one that is being measured. What is being measured here would be the plant growth. So you measure it in height, number of leaves, etc. So you're measuring whatever it is that shows growth on the plants. Then what are we keeping constant? This is our controlled variable. So for this experiment, maybe the type of plant is being kept constant, or the type of soil, the amount of water given to each plant, and the temperature. So that is how you do it. So you read the statement and then you break it down into the independent, dependent, and controlled variables. Let's try some by ourselves now. Good luck! 2. A researcher studies whether increasing the amount of fertilizer affects the size of tomatoes. So again, you're going to look at this statement, you're going to read it, you're going to understand it, and now you have to determine what are you going to change purposely, what are you measuring from this, and what are you going to control if you were to do this experiment. Yay, did you get it? Was it close? Was it different but correct? Very good. So the independent variable here will be the amount of fertilizer. Dependent will be the size of the tomato. And then we have the control, so the type of tomato plant, the amount of sunlight exposure, and then the watering schedule. Here's number 3. A student investigates how the temperature of water affects how fast sugar dissolves. Come up with your independent, dependent, and controlled variable. Awesome. So we have the temperature of the water being the independent variable. Then we have the time it takes sugar to dissolve, which is the dependent variable. And then we have the controlled variable being the amount of sugar used, the volume of water, or the stirring method. 4. A teacher measures how different study times impact students' test scores. Give it a go. Alright, so the independent variable here would be the duration or study time. Dependent would be the test scores. And then what are we controlling? The group size, the level, the intelligence level, the type of tests, the age of the students, the test difficulty, and the test environment. So a lot of things you can control here. Number 5, a chef experiments to see if the baking time affects how soft a cookie is. What do you think you would have as an independent variable? And what do you think you would have as a dependent variable? And what is the control variable here? What are you going to purposely change? What are you measuring? What is responding here? And what are you keeping controlled or constant? Awesome. So the independent variable will be time or duration. Dependent will be the softness of the cookie. And then we can control many things: the ingredients, the oven temperature, the type of cookie dough, and the cookie recipe. 6. An athlete wants to know if different stretching routines impact sprinting speed. What are we going to purposely change here? What are we going to measure here? And what is constant? Great. Types of stretches are stretching routine. Then that is for independent. Then we have for dependent is how fast the athlete runs or sprinting speed. And then the constant variables here will be that each time we use the same athlete, the type of track being run on, our track conditions, the distance being covered, and the athlete's diet and the weather. A scientist examines how different salt concentrations in water affect the hatching of brine shrimp. What are you going to manipulate? What is being measured? And what is being held constant? Awesome. So the independent variable would be the amount of salt or salt concentration in the water. The dependent variable here will be the number of hatched brine shrimp or the hatching of brine shrimp. And then what is being held constant would be the species of brine shrimp, the volume of water, the temperature of the water, or the type of salt used, and the light exposure. So any of those would work. 8. A student investigates whether the color of light affects the growth of algae. What is being manipulated? What is being held constant? Awesome. The colors of lights used would fall under the manipulated variable. The growth of the algae, the height, the number of leaves would fall under measured or responding, which is dependent variable. What is being held constant? Temperature of the water, the light intensity, duration of light, species of algae, quality of water. So if you came up with any of these things, very good. Awesome sauce, students. All right, number 9, a gardener tests if the frequency of watering affects how quickly grass seeds germinate. What is the independent variable here? Remember, the independent variable is the one that you purposely change. What is your dependent variable? What is the one that's responding? What's being measured? What are you controlling? What is being held constant? Very good. The frequency of watering would be the one that you're manipulating. The time taken for grass seeds to grow would be the responding. So remember, dependent is either the one being measured or the one that is responding. And then control, what is being held constant? The amount of water, the quality of water, the type of water, type of grass seeds, soil type, sunlight exposure. And finally, the last one, a researcher wants to find out how the concentration of caffeine affects heart rate in adults. Good luck! Awesome sauce, everybody. Did you get something close to this or exactly like this? You're doing a great job. So we have concentration of caffeine being independent, the heart rate being dependent, and what is being controlled? brand of caffeine, duration of experiment, age of participants, amount of physical activity, and time of day. Great. So you do know that you may have to work in groups. Some of us don't like that, but that is life. So when you start on a project, you must be clear about the question you are investigating. And as a group, you should discuss this together and sort out the different aspects of the problem. Then you can divide the work between the members of the group. That's how group work goes. So guess what? We have a fun FUN activity for planning and designing a lab. You were introduced to a planned and designed lab earlier in this series. So now you have to actually do one yourself. You can pick any of the ones listed below, and you will be working in groups. So are you going to pick A? Are green fruit that are wrapped in newspaper ripen faster than unwrapped green fruit? B. Are objects that are painted black dry faster than objects that are painted white? C. A scientist is testing whether plants watered with rainwater grow faster than those watered with tap water. D. A gardener is trying to determine if using organic compost results in larger vegetables than using chemical fertilizers. E. A scientist is curious to see if colder temperatures slow down the rate at which bacteria grow on food. F. A student is trying to determine if studying in a quiet environment leads to higher test scores than studying in a noisy environment. So what is it going to be? A, B, C, D, E, F. Pick your choice. Have fun. Use the skeleton for writing up a plan and design on the next page. So this is what you would have seen if you saw the earlier parts to this series. It's the same thing. So you have to write your name or the names of group members. So you write your lab number, the date, problem, the hypothesis. Remember, we learned how to do a hypothesis already. You're going to write your aim. What is the point of you doing this? Come up with some material and apparatus. I would suggest that you do probably the method first, or you do your variables first. So if you do your variables first, you could come up with your materials and apparatus next, because then you would know what to do, and then you could come up with your method. From there, once you do all of that, you should have an idea of how you're going to tabulate your results, and then you can either pick up an assumption, a precaution, or limitation. Okay, good luck. What are an assumption, a precaution, and a limitation? An assumption is something you believe is true in your experiment. For example, the ruler that was used may not have been accurate, while a precaution is a careful action you take to keep the experiment safe and fair. For example, goggles should be worn when heating a liquid. Lastly, a limitation is a problem that makes your experiment less perfect. For example, only one type of plant was tested, not many kinds. Examples of assumptions. The assumption or assumptions that are written for the lab being planned and designed should match the context of the lab. Here are some examples of assumptions below. The scale may not be properly calibrated. The acid may be contaminated. The seeds may not be from the same species. And the timer may not be properly calibrated. Examples of precautions. The precautions or precaution that are written for the lab being planned and designed should match the context of the lab as well. Here are some examples of precautions below. Handle the scale with care. Handle the acid with care. Store the seeds in a cool, dry place. And handle the timer with care. The same thing goes for the limitation when we're talking about context. Here are some examples of limitations below. A poorly calibrated scale may cause the documentation of inaccurate results. The age of the acid may cause the documentation of inaccurate results. The quality of the seeds may cause the documentation of inaccurate results. and the poor reaction time when handling the timer may cause the documentation of inaccurate results. Tense. Plan and design lab reports are always written in the present tense because the instructions telling what to do in the future, while lab reports, on the other hand, are always written in the past tense because they're written after the experiment is completed. They include the actual results shown in tables or graphs, comparisons with literature values or peers' work, and explanations. Thanks for watching. See you for part 7.